Hello, everyone. Welcome to our webinar that's offered jointly by the Mobilize Center and Restore Center at Stanford University. My name is Carmichael Long, and I'm a research engineer with our centers, and I'll be serving as the moderator for today's webinar. I'm pleased to welcome today's presenter, Kevin Thomas. Our webinar today is divided into two parts. In the first half, Mr. Thomas will describe his research on deep learning models for analyzing images for osteoarthritis. And in the second half, he'll cover best practices for training deep learning models. Today's webinar series is brought to you by the Mobilize Center and Restore Center, which are both supported by the National Institutes of Health. The Mobilize Center is focused on developing and disseminating state-of-the-art biomechanics and machine learning tools for researchers to analyze human movement. The Restore Center is working to make these and other tools for real-world assessments of movement more widely available to the rehab research community. Before we get started, a couple quick reminders about the format of the webinar. We will have a research talk and then a discussion on best practices, and we'd love to take your questions. We'll take some questions both at the end of the research talk and at the end of the second half, and we ask you to type those into the Q&A panel, so not the chat, but the Q&A panel. So now we'd like to go ahead and introduce our speaker for today. Kevin Thomas is an MD PhD candidate at Stanford University, completing his PhD in biomedical informatics under Scott Delp. His research focuses on the development and application of machine learning methods for human mobility data. Before joining Stanford, Kevin received a BSc in biomedical engineering from Arizona State University and conducted research at the University of Calgary Human Performance Lab as a Fulbright scholar. So with that, I'm very excited to have Kevin and all of you joining us today, and I'll let Kevin go ahead and begin.
Thanks, Carmichael. Yeah, let me shift over. Okay, yeah, thank you to everyone for tuning in today. Osteoarthritis, or OA, is one of the most likely causes of knee pain in older adults. It involves inflammation and degradation of the structures of the knee, including the bones and the cartilage. Now, medical imaging allows us to visualize the tissues that make up the knee and assess how healthy they are. And for that reason, imaging is an important source of structural disease severity measurements in OA research. And whether we're using X-rays or MRIs, measuring joint health for medical images usually requires someone to look at those images and identify signs of disease. This is a time-consuming task, and it requires a lot of training to do properly. And even among experts, the intra-rater repeatability and the inter-rater agreement are imperfect. In other words, you might show two radiologists the same image, and their evaluation on how healthy the joint looks often differs by some amount. And while it's not as subjective as relying on patient-reported pain, subjectivity among readers reduces researchers' ability to detect true changes in health in longitudinal clinical trials. This makes it necessary to run longer trials with larger cohorts. And at the same time, the cost and expertise required to perform these assessments reduces the number of research groups that have access to objective measures of structural disease, and that hinders the assessment of large cohorts. Now, in order to relieve this bottleneck, it'd be ideal to automate the assessment of OA severity from these images, and deep learning might be the key. Deep learning is a computational technique used to create neural networks, which are pieces of software. And these neural network models provide a mapping from an input to an output by learning from example data. So you give a deep learning model an input, and it gives you back an output. For example, we might want a model to learn how to take in X-rays as input and produce an assessment of OA severity as output by showing it example images that are previously labeled with OA severity by a radiologist. However, deep learning models often only perform well on new on-scene data when it falls within the distribution of the training set. This means that a model trained on images from a research study at one site might not perform as well on images collected in clinical practice at a different site. For example, here's an image from a site whose images have inverted pixel values because of the machines they use. If our model never saw an image like this during training, it might not produce an accurate assessment for it. And before using models in practice, we need to test them on diverse set of patients from a diverse set of research studies and clinical sites to make sure they perform well in real life. And these tests need to go beyond traditional machine learning validation assessments and evaluate models in the way OA researchers are actually going to use them. Now, there are many prior works that have automated various OA-related assessments from X-rays and MRIs using deep learning. But unfortunately, the majority of them haven't made their models publicly available. And even when they do, it's often in a form that's difficult for OA researchers to use. And when OA researchers can't test the models themselves, the models often don't get validated on diverse data sets. And this prevents them from being used in the real world to relieve the medical imaging bottleneck that's currently hindering OA research. So today, I'll talk about work we've done to improve upon the previous state-of-the-art for deep learning models that automate OA imaging assessments. I'll talk about validation tests we've done to justify their use in clinical research, and also about how we've deployed them in a way that enables external validation and use by OA researchers. The first project I'll talk about focuses on automating one of the most commonly used scoring systems for measuring OA severity in X-rays. And the second project I'll talk about focuses on automatically finding the cartilage in compositional MRIs and using that to track subjects' joint health over time. And in this first project, we focused on automating what's called the Kellgren-Lorentz scoring system, or the KL scoring system. And this is a discrete 0 to 4 score of osteoarthritis severity based on X-ray findings. And this is stuff like joint space narrowing, or osteophytes, which are also called bone spurs, sclerosis, which is also called bone hardening, and deformity at the ends of the bones. And in the KL score, a 0 denotes a healthy, normal joint. And as you move from a 0 to 4, you see more and more evidence of cartilage degradation and abnormal bone remodeling. And in order to train a model to automate the KL scoring system, we used 20,000 bilateral X-rays from the Osteoarthritis Initiative dataset. For each image, we split it vertically down the middle so that we now have 40,000 single knee X-rays. And we split these images up at the subject level so that 80% of the subject's images went into our training set, 10% went into our validation set, and 10% went into our test set. And splitting it up like this allows us to get a more robust, rigorous assessment of how well the model might perform on new data it hasn't seen during the training process. Now, when we talk about neural networks, a model's architecture determines its strategy for how it maps an input to an output. And we used the 169-layer dense convolutional network, also called the dense net. And we modified the final layer so that it would only have 5 outputs corresponding to the 5 different Kellgren-Lorentz scores that an image might have. And each of these 169 layers builds on information from the previous layers in the network to detect visual patterns from the X-rays. So as you progress through the layers from the original input image through to the diagnosis at the end, you progressively get visual patterns detected that are more abstract and more specific to the task of estimating KL scores. Now, the unique thing about the DenseNet architecture is that each layer directly receives information from all of the previous layers. This means that the final layer, which is responsible for actually predicting the KL score, still has access to that fine-grained, detailed information of the original input image. And if we think about the subtle patterns that differentiate sclerotic bone from normal bone, this intuitively feels like a desirable trait for our model. Now, ultimately, our model achieved state-of-the-art results. And to assess our model's performance, we looked at how well it agreed with a committee of radiologists relative to how well individual radiologists agree with a committee. And there are several different metrics you can use to assess the agreement between 2 raters or between a rater and a model. The first one we used is called the F1 score. And technically speaking, this is the harmonic mean between precision and recall. But in more simple terms, what this means is that we want a model that both accurately detects when arthritis is present and also is able to recognize when it's not present. And the F1 score balances both of these priorities into a single number between 0 and 1. Another metric we used is called the Cohen-weighted kappa. And this gets at the idea that if a committee of radiologists agrees that a certain image should have a KL score of 4, it's better for our model to assign it a KL score of 3 than a KL score of 0. So Cohen's weighted kappa accounts for the fact that the KL scoring system is a numerical ordinal system. And as you move from 0 to 4, you're progressively getting more and more severe disease. And the last metric I'll show today is accuracy. And this is just, simply how many images the model is correctly labeling. But it can be a little bit misleading for some data sets because for our osteoarthritis initiative data set, for example, there was some data imbalance, meaning we had a different number of images in each of our KL scoring categories, which can shift the priority so that accuracy isn't a balanced metric of performance, but it's intuitive, so I'll show it here. And when we look at how well our model agreed with the committee relative to how well an individual radiologist agreed with the committee for a 50-image subset of our data, our model outperformed the individual radiologist in all 3 of these categories. And when we look at how well the model agreed with the committee of radiologists for our full test set of thousands of images from hundreds of subjects, we performed with state-of-the-art results in all 3 of these performance metrics. Now, after we confirmed that our model is good at assigning Kellgren-Lawrence scores, we wanted to confirm that it's using clinically relevant information to obtain these KL scores. So after we used forward propagation to work our way through the network and process our image through the layers of the model to get a KL score, we then used backward propagation to work our way from its output back to the pixels of the original input image. And this produced something called a saliency map, which can be thought of as a heat map that identifies the relative importance of each pixel in the input image to the model's final prediction. And these saliency maps confirmed that the model is using clinically relevant information. These saliency maps consistently highlighted the medial and lateral joint margins and the tibial spines here, which are common locations of osteophytes, which you'll remember were one of the big important factors that experts look at when they're assigning Kellgren-Lawrence scores. And if you look at the middle image here, you can also see that it highlights areas of joint space narrowing, which is another important factor in the Kellgren-Lawrence scoring system. So after we validated that it performed with state-of-the-art results and is using clinically relevant information, our next step was to deploy the model in a way that enables OA researchers to actually use it. So we've used 2 different deployment strategies here. First, we've released our model as a Docker, which allows researchers to process a large number of images with just 3 simple command line arguments. And this is available at the URL here on GitHub. And we've also produced a web app to make it easy to use. And this is available at kl.stanford.edu. And it allows researchers to simply drag and drop images into their browser to have them analyzed automatically. Now, a lot of researchers aren't able to share their images with outside research groups because they need to protect their patients' privacy. So we designed our web app so that no information ever leaves the user's computer. All processing is done locally, and users can even turn off the internet while they're using the web app and still continue to process their images. Now, when imaging is done to assess OA severity, it's usually X-rays because they're affordable and fast. But the drawback of relying on X-rays is that these assessments are based on bony deformation and joint space narrowing because cartilage isn't visible in these images. So by the time we can see signs of OA on X-rays, it's likely that the disease process has already been going on for quite a while and affecting the cartilage. Because bony changes happen slowly over many years, X-rays also aren't ideal when we want to efficiently evaluate potential interventions over shorter periods of time. But because MRIs directly visualize the cartilage, they offer the potential to detect OA-associated changes sooner. Compositional MRI sequences are used to delineate the biochemical or microstructural properties of cartilage. Now, in these images, each voxel or 3D pixel that displays cartilage also gives you a score for how healthy that small region of cartilage is. This score is based on factors like the content of collagen and proteoglycans and water in the cartilage. So one example of a compositional MRI sequence is the multi-echo spin echo T2 weighted MRI, but that's kind of a mouthful. So I'll just call that the MESI MRI for multi-echo spin echo. Now, this is one of the most commonly used accessible sequences for compositional MRI. And the numerical score for cartilage health that MESI MRIs provide is called a T2 value. Now, T2 values tend to increase as OA progresses, and these T2 increases are thought to reflect the collagen breakdown and changes in cartilage hydration that are seen in osteoarthritis. Now, the challenge with using MESI MRIs to assess cartilage health is that you first need to find where the cartilage is within your image. Now, this process is called segmentation, and it's usually done manually or in a semi-automated process by an expert, and either way, it's very time-consuming. So efforts to automate cartilage segmentation have mostly focused on structural sequences used for morphological assessments like measuring cartilage thickness. But automating the segmentation of MESI MRIs is crucial for assessing early changes in cartilage matrix composition at scale, so that this imaging modality is more feasible for researchers to use. So our goal was to develop a neural network model that takes in MESI MRIs as input and generates accurate segmentations that are good enough to track longitudinal changes in cartilage T2 values. And the data we used, again, came from the Osteoarthritis Initiative dataset. We used 2 longitudinal MESI MRIs, one taken at baseline and the other taken 4 years later from the right knee of 143 subjects without radiographic OA. And each MRI was segmented by a musculoskeletal radiologist with 15 years of experience. And I'll refer to that radiologist as reader 1. We then split the subjects into a training set, a validation set, and a test set. And one image from each of the test set subjects was also manually segmented by a second expert, who I'll call reader 2. And we did this to see how well the experts agree with one another relative to how well the model agrees with each expert. And we used a neural network with a 2D UNet architecture. This means that the model first summarizes the relevant features in each MRI slice into a summary vector here, and then uses that summary to estimate where the cartilage is located in that slice. And to validate the model, we first tested each test set image and looked at how well the segmentation mask from the model overlapped with the segmentation mask produced by the expert. Then we calculated the T2 value for each segmented cartilage voxel. Once we had that, we projected the 3D femoral cartilage onto a 2D plane. You can think of this process like peeling the cartilage off the end of the femur and then laying it flat on the table. We're able to do this computationally by fitting the segmented cartilage to a cylinder using least squares, and then unrolling the cylinder by re-expressing the location of each cartilage voxel using polar coordinates. So for each cartilage voxel, we know which slice of the MRI it's located in. And from the polar coordinates, we also know where along the arc of the cylinder it's located. And we use that as the y-axis location. And then we color code according to the intensity of the T2 value. After we have these 2D T2 maps, we then automatically divide the cartilage into 12 subregions. And these 12 regions were defined by drawing boundaries between the superficial and deep halves of the cartilage, between the medial and lateral halves, and between the anterior, central, and posterior thirds of the cartilage. And then we compared the model's estimated T2 value and T2 changes over time with those metrics obtained using expert segmentations for each region. And when we compare the cartilage masks produced by the model to those produced by our expert, we use a metric called the DICE score. This measures how closely 2 segmentation masks overlap, and a value of 1 indicates perfect agreement. On average, our model agreed with reader 1 with a DICE score of 0.85 for our full test set of images. And on the 14 images that were also segmented by reader 2, our model agreed with reader 2 with a dice score of 0.75, while reader 1 agreed with reader 2 with a dice score of 0.74. So overall, the model segmentation masks agree with each expert as closely as the experts agree with one another. Now, we also then looked at how closely the model's estimated subregional T2 values agreed with those of the experts. And so I'm going to show a number of plots like this. So first I want to just walk you through it. On the left half, that represents the medial half of the femoral cartilage plate. And the right half of these plots represents the lateral half of our cartilage. And the number at the bottom indicates the agreement between our model and reader 1 for the full cartilage plate. And for this first slide, I'm going to specifically be focusing on the superficial cartilage. And for estimating T2 values, our model agreed with reader 1 extremely closely, where we had a Spearman correlation of 0.75 or higher for each of the smallest subregions, and a correlation of 0.91 for the full superficial cartilage plate. And the agreement is even better for the deep cartilage plate, where our model agreed with the radiologist with a Spearman correlation of 0.87 or higher for each of the smaller subregions, and a Spearman correlation of 0.97 for the full deep cartilage plate. Now, we also wanted to look at how well our model could measure changes over 4 years in the cartilage. And our model continued to perform well on this task as well, where we saw strong Spearman correlation coefficients in all major subregions of the superficial cartilage. And they were all statistically significant except for the superficial anterior lateral subregion, which had a p-value of 0.08. And again, for the full superficial cartilage plate, when we're tracking changes in T2 values over 4 years, our model had Spearman correlation with the experts with a value of 0.88. And again, we continue to see strong results for the deep cartilage when we're tracking changes over 4 years, where our model had a Spearman correlation of 0.70 or higher in all of our small subregions, and a value of 0.96 for the full deep cartilage plate. So overall, the model segmentations are good enough to track changes over 4 years, which we were really excited about. Now, our ultimate goal is to provide a validated tool to the research community that can actually be used to accelerate OA research. So we've made our trained model publicly available on GitHub at this URL, along with the code necessary to use this model on new images. And we've also made a web app again to make the model even easier to use. Similar to our X-ray web app, researchers can go to kl.stanford.edu to simply drag and drop an image into their browser to have it analyzed. It only takes a couple of minutes per image on a standard laptop, and it provides the option to view and download those segmentation masks. Again, with all processing happening locally on the user's own computer, so you don't have any need to share data. So overall, we've developed, validated, and deployed models to enable researchers everywhere to obtain quantitative, objective imaging biomarkers without the bottleneck of manual image annotation. With that, I'll pause for a minute to take questions on this first half of the talk.
Great, thank you so much, Kevin. That was a great talk. So now we'll go ahead and start the Q&A session. If you have a question, please go ahead and type that into the Q&A panel, not the chat, the Q&A panel, and we'll get to as many questions as we can. We'll probably take about 5 or 10 minutes, and then we'll switch over to the second half. So we've already got one question in from Priyanka Agarwal. Nice presentation. How much time per X-ray image / MRI is needed roughly for the output via deep learning? Has this method been used for managing critical issues like bone necrosis and where time is of essence to reestablish blood flow?
Yeah, that's a great question. So for X-rays, the model is able to process those images in less than 10 seconds on a standard laptop. And for MRIs, usually it's about 3 minutes on a typical laptop. But in both of those cases, there's the opportunity to accelerate that processing time by using a computer with a GPU or a graphical processing unit. And so even in, I think, really time demanding contexts, these can be really helpful, have a lot of potential.
Great. I guess I'll ask some questions while we're waiting on some other questions to come in. Some interesting results out of the MRI section between, I thought, the superficial and the deep. Do you have any clue as to why it looked like some of the deep cartilage numbers were a little better than superficial? And is it more important to get either one of them right, or are they kind of both equally important?
Right. So I think overall, with the exception of that one superficial lateral anterior region, the model's ability to track both T2 values at a given cross-sectional image and track them over time were really strong for both the superficial and deep. And I think they're good enough to be useful in practice for the full cartilage plate, both superficial and deep. But you're right that the numbers were a little better for the deep. And I think part of that relates to how the disease process carries out in osteoarthritis, where the cartilage starts to undergo degradation at the superficial surface first, and progresses its way from the superficial surface more deeply. And so that kind of textural roughness in the cartilage at the surface makes it a little bit trickier of a segmentation task, and allows, I think, for just a little bit more of a reliable segmentation for the deep. But yeah, no, you're completely right that, yep, there is a little bit of difference. And I think those are also challenges that human experts face too when they're trying to really figure out what that boundary line is between the superficial cartilage and the outside of the joint.
Yeah, absolutely. I thought it was nice that the correlations were kind of in line with human error. So it's almost as good as you can kind of get, maybe, to a certain extent, since that's what you're comparing to. Is that kind of right?
Right. So whenever you're developing a new machine learning model, it's nice to have a benchmark of performance and to have some estimate of what the ceiling of possible performance could be. And so oftentimes for neural networks for medical imaging applications, you try to figure out, how well do human experts agree with one another when they're trying to assess them. That's oftentimes your benchmark for, okay, like when we hit somewhere in that ballpark, we're reaching the high end of the theoretical possible performance that we could get with a model like this.
Cool. We got a couple more questions in. So from William Anderst, does the resolution, pixel size, and slice thickness for MRI have to match the training image resolution? Has the software been validated using different resolutions for CT and MRI?
Yeah, so you're exactly right. When a model is trained on a certain data set, it's usually designed to perform well on the certain parameters with which that type of medical image was captured. So when we're specifically talking about the resolution of a given slice, if there's ever any MRI that's produced with higher resolution than what we trained on, it could easily be downsampled so that it would be at the same resolution that our model was trained on. And for our MRI project, the slices that our model was trained on had only 384 voxels by 384 voxels, which is a relatively low image resolution, which means that most of the images you might capture in practice would be at least that good. And then you could therefore downsample it to get it ready to feed into our model for segmentation. In terms of slice thickness, I think that's an area we haven't done as much validation work as I'd like. We were restricted to the Osteoarthritis Initiative data set, which had a pretty homogeneous slice thickness. But one of the beauties of making our work publicly available and open for others online is that more of this validation work can happen at other sites that have other imaging machines with different parameters. So I'm looking forward to hopefully doing external validation studies and potentially collaborations down the road so that we can really pinpoint, how well our model does on a broader distribution of data. That's a great question. Thank you.
Thanks. And then one more from Priyanka Agarwal. How does a clinical person wanting to use this app know if the images available to them are appropriate for the deep learning, to work on them?
Yeah. So for these two apps, I think that's a really important question because, like I said in the talk, the models typically only perform well on the type of images they were trained on. So I guess broad picture in general, when you're trying to assess any machine learning model and whether it might be useful to you in your own applications, one thing would be to look up the publications that are available online. And so like for our X-ray paper, we've published it, and the MRI paper will be coming out very soon. And it gives a detailed description of the specific type of medical imaging that was used to train the model. So for our X-ray project, these were AP view fixed flexion knee X-rays, which means each knee that it saw during training had a pretty fixed amount of knee flexion when the X-ray was captured. And then for the MRIs, our paper will describe all of the imaging parameters that were used in the model. And you can also find that information in the documentation of the Osteoarthritis Initiative data set. And then the next question is to identify, are my images pretty much in line with the images that were used during training?
Thanks. So I think we're going to want to leave time for the second half on the discussion of best practices. So we'll go ahead and switch over to the second part, and hope that everyone will stay with us.